Good morning, everyone. I, Dr. Sakshi Bhardwaj, on behalf of Feltham Technologies, welcome you all on our Women's Day special webinar series. Today, I am here with a new and extremely fascinating topic that is entitled as single cell RNA sequencing and the bioinformatic analysis pipelines. We all know that the development of next generation sequencing, which is the NGS technologies, has developed quickly in the recent years. And this has given rise to numerous useful insights into the complicated or complex biological systems, from the genetic of cancers to the various microbial communities. The characterization of individual cells is becoming more and more of focus for NGS based technologies in genomics, transcriptomics, and epigenomics as well. For instance, single cell RNA sequencing can identify complex and uncommon cell populations, explain how genes are regulated, and trace the development paths of many diverse cells. Our expert of the day will be talking about how single cell sequencing is a powerful tool that can provide a more detailed understanding of the cell populations within a tissue or a disease, which can help to identify novel therapeutic targets and improve the efficacy of existing therapies. It's giving me immense pleasure to introduce our guest of honor for the session, Dr. Retha. She is a postdoctoral associate at the Hen and Robert H. Laurie Children's Hospital of Chicago, United States. She has more than 5 years of experience in the field of computational biology, healthcare, and life sciences. She has her expertise in data science, machine learning, natural processing language as well. She is well versed in next generation sequencing, electronic medical records, along with hands on experience in the molecular biology and immunotherapy techniques. She has done her MS in Health and Biomedical Informatics, the Feinberg School of Medicine, Northwestern University, United States, PhD in Biotechnology Chemistry, University of Madras. She had been a research scientist at Ashtagiri Herbal Research Foundation, Chennai, India, and visiting researcher in David Geffen School of Medicine at the University of California, Los Angeles. She had been involved in various research projects and got awarded with various fellowships too. She has several national and international publications along with the four patents. Now it's time to hear out our guest speaker. Over to you, Dr. Rita.
Thank you so much, Shakshi, for the humble introduction. Good morning, everyone. My topic will be on the single cell sequencing and the bioinformatic analysis pipelines. I'll start with what is the next gen sequencing and what is RNA sequencing. So, to start with RNA sequencing is nothing but isolating the RNA and sequencing it to see what are the cell types and what are the genetic markers which can be analyzed from a disease or a tissue. So for RNA sequencing, there are two types. One is bulk RNA-seq and the other is single cell RNA-seq. So for bulk or population RNA-seq, you isolate the RNA material from the tissue or the cell, and you do an sequencing where you get an average gene expression for all the cells in a sample. But whereas for single cell RNA-seq, it's a little bit more complex, but it gives you a more defined objective of what the single cell is, not just the genetic marker, but the genetic marker for each single cell or a transcript. So this is kind of a more updated version of the bulk RNA-seq. And in this, you would be able to analyze more of not just the genome, but also the cell types and the antigens and the specificity, the protein specificity of each of the cells. So it's an amazing technology, and which kind of has transformed the way we look at molecular biology and diseases. So to start with single cell, For single cell, there are multiple methods which people have started using, like single cell RNA sequencing, single cell DNA sequencing, single cell epigenetics, single cell proteomics, and single cell 10x genomics, and all. So let's not get into the other single cell analysis. We'll just stick with the single cell RNA-seq, which is going to be the 10x single cell. Why I'm choosing 10x is there are other technologies like SmartX, SmartSeq, single cell, DropSeq, and all those. But the most widely used now and the less expensive one is the 10x genomics. So I'm going to start with saying how the 10x genomics works and what are the advantages and what can we do with 10x genomics. So 10x Genomics is just the company name, which and the technology behind that is a droplet seq technology. So they use droplets to kind of prepare the whole single cell with the other antibodies and the proteins and the antigens. So if you could see the image on the right, it is an image which has RNA, proteins, T cell, B cells, CRISPR, and chromatin. So with single cell, you can either do single cell sequencing or single nuclei sequencing, which is the same process, but instead of the single cell, you're going to lyse the cell and take the nuclei out. There is another technology also, which is the chromatin, single cell chromatin sequencing, but we're not going to dive deep into that. I'm just saying the various single cells which are there. And one more interesting thing is single cell has developed so widely in the last 2 years that along with single cell, you can also do TCR and BCR sequencing and also CRISPR screen knockouts. You can add your CRISPR screen along with your gene expression profile. So that's kind of fascinating. Um, so as I told you, they're different assays. But what is for all these different assays, what is the input material you need? It's still RNA, but for the RNA, you need, in case you need to do the protein expression with the RNA, which is called site-seq RNA. So with your single cell, you will be doing site-seq analysis. So the terms might be a little confusing, but I'll just make it very simple. So site-seq is nothing but you have surface marker proteins on the cells. So instead, not just the RNA is going to be analyzed, you're going to even try to analyze the surface markers to see what are the receptors present on the surface. So this can be done with the whole cells. And then you have just the gene expression where you don't do the surface markers. For that, you just need the RNA. And you could do this either on the whole cells or the nuclei, which is after lysing the cell. You have a third category, which is the RNA and the chromatin. And for this, you just need the nuclei. You don't need the whole cell. So you can lyse the cell. You can take, uh, uh, after lysing the cell, you can take the RNA and you can do the chromatin analysis. The ones which we are concentrating now is going to be the one in the middle, which is the gene expression. And I will dive into the other two little bit just to give you a glance of what the other two are and how you can utilize that in your research. The main challenges when it comes to single cell experimental design is the number of cells you capture. Like, it's not very easy to get the amount of cells you require for this sequencing because you're going to deal with preclinical and clinical samples. So when you say clinical samples, you're not going to get much sample. It's going to be a small, tiny pinch of tissue from any patient. So it's going to be very less number of cells. So that's kind of a very challenge. The other is the sequencing depth. So the more the sequencing depth you need, the more expensive it becomes. So that's kind of another challenge which we need to look into. What is the sequencing depth we are looking at? And then isolating single cells. So when you're desolating your tissue, it's very important that you get single cells and you leave out the debris and the artifacts. So that's also a challenge because it's really hard to kind of remove the RNA, ambient RNA, and the debris from your cells. And the fourth one is the RNA extraction and the library prep. So though there is a protocol, you have to be very stringent about how you do that. You need to, once you extract your RNA, you need to know your DV200, whether it lies within the threshold of at least 80% purity. If it is not so, then I don't think you would be able to perform the analysis. And the next one would be, once you have your RNA, you would convert it into cDNA, and you need the steps for amplification. So this, you need to know how many amplification cycles is needed. Then after that, you go to the library prep, and again you have another challenge there where you have to saturate your library. After that, it's again the experimental noise and debris, which is kind of the data analysis part, and the sparsity of the data and the batch effect. All this comes for the data analysis, but again, you need to take into consideration all this. Batch effect is one important thing which needs to be noted, because you might have 8 samples once, and then you might run another eight samples. So there is a batch effect between both the samples because it's not run in the same time. And since if it's a preclinical, maybe you can like try to process it all at a time. But when it comes to clinical sample, you cannot control the patients coming in, right? So that kind of gives you a batch effect. And during data analysis, you can try to control for all these challenges. Okay. Uh, Let me start with the overview of what 10x genomics, 10x chromium single cell technology is. We call it 10x genomics. And I'll just go into what the technology is. So to start with, the picture which says A is nothing but how the whole process is designed, the flow. So to begin with, you get the cells. Once you have the cells, you add the beads. The beads is nothing but the gem beads, which is the droplets into which the cells go and settle down. And then you kind of put this into a chip plate, which is actually the sequencing chip plate. And then you put it into a chromium controller, which mixes up the sample and gives you uh, a, a sample which is embedded into the droplet. So each of your cell will be embedded into a droplet with the other libraries which are added into it. And then you get the gem beads, you do an RT-PCR, and you convert the RNA into cDNA. And after this, you amplify the cDNA, and then you construct the library. And once the library is prepared with whatever you want to put, the proteins, the T receptors, the B receptors, then you can directly take this whole gem bead for sequencing. So this is the first step. Looks simple, but there's plenty of other steps which follows this. So when I said the gem bead is nothing but actually a primer, like a RT primer within a bubble. So what you do is you add this primer into the reagents, and you make a droplet with the oil. And then finally you collect the droplets as single cell gems, which has the antigens, which has the antibodies, which has the cells, everything embedded into a single droplet. So if it has multiple cells within one droplet, you're going to have an issue. Like it's going to have a doublet, which is going to give you a wrong number of biomarkers when you do the analysis. So it's very important that you have only one single cell in one gene bead. Uh, you can check the quality of the gem beads, and if you can see anything which is zero with a number of zero beads would not be recommended, and anything with a number of gem beads of one would be the best bet you have. And this is just a result which shows that there was 80% of gems which were with a single cell in it, and it's an amazing result. So as I told you, we do the RT-PCR and all those things. So this image is nothing but telling you what is that we add. So the 10x barcodes are just barcodes for each of the sample to recognize, because you're going to add multiple samples into one sequencing chip. So you add barcodes into them, and you have UMIs, which is unique medical identifiers. So this tells you that which sample is belonging to which patient. It's You can say that these are just like barcodes, because when you add a mixture of samples and you're doing a lengthy sequencing, you need to know from which cell or which sample the whole cDNA is coming from. So these are just unique barcodes which you add in the beginning, and then you do the RT-PCR and you get the processed cDNA with the barcode. The next would be, uh, after you do the cDNA and then the amplification, your final sequencing would be something like this. You'll have a barcode, you'll have a UMI, you'll have the oligonucleotides, you'll have the cDNA, you'll have your sample index. They do so again with single cell. Earlier, like 5 years ago, maybe there was just 3' analysis, where from the 3' end they used to do the sequencing. But now there's something called 5' sequencing too. So you can do both the sequencings. Like you can try to do the sequencing from the 3' end and also the sequencing from the 5' end. The last final step after you do the sequencing is your data analysis. So you extract the barcodes, the UMIs, the RNA reads. You correct them with the barcodes and see the sequencing. And this is all done with a tool called Cell Browser. Or you can do it by yourself using the usual star, top hat, or any other tool which you use. You aligned it with the star, which is your reference genome. Then you see what are the tags or the transcripts. You correct in case you have any missing UMIs, you correct that. And again, the Hamming distance is nothing but the sequencing from one nucleotide to other, and it should be one. So that's kind of like a norm. And then you count the UMIs, and then finally you have the gene matrix, which is actually the gene and the count for each of the cell. This would be the final step where you go for the downstream analysis. So when you go for sequencing, this will be the results you get. Diving little into the next gen technology, I told you that you can do various things with sequencing. So the two things I emphasized on was nuclei and cells, but other than that, there's something called CRISPR guides. You can add your RNAs, like CRISPR guides, knockouts into the gel beads, and also you can add your barcode antibodies. Barcode antibodies are nothing but your surface markers. Your cells would have surface markers. Suppose you're looking for a T cell, it would have a CD4 receptor. So you could add the receptor into the gel bead and you can quantify it later. The last one is barcoded peptide MHC multiers. It's nothing but the T cell and B cells specific antigens. So you can add these B cells and T cell specific antigens to see what the subtypes of the T cells are, or what the VDJ chains are. So that's a very interesting study because people have dived more into T cells and B cells. And single cell has given a payway for this type of analysis. Before we could get into the data analysis, I know some of them might have the question of how do you prepare the sample, how is it done, and what is the procedure. So basically, whether it's a preclinical sample or a clinical sample, the most important thing is the tissue collection. So you cannot have a tissue which is collected like 72 hours later. So when you have a tissue which is collected 72 hours later, the cell viability decreases and also the cell count decreases. So it's very important how you collect the sample and how the sample is stored. Whether it's a mouse sample or whether it's a human sample, it's so important that you store the sample. And we have few techniques which generally is followed, like snap freezing or hypothermic tissue preservation or direct tissue preservation. I'll tell you how it is used and what are the considerations for each of it. So for tissue storage solution, it's a fresh method where you snap freeze you get the tissue and you snap freeze it in nitrogen. So this is within 72 hours, and you can transport it using ice packs or cold packs. But after 72 hours, the tissue becomes obsolete. You cannot use it because you're not going to have any cells which are viable. So that's kind of a tricky situation. The next thing would be flash freeze the whole tissue. And this is kind of a long term storage, which is like more than 72 hours. You put it in minus 80, or if you need to ship it, you ship it with dry ice. Then your tissue is fine and your cells are still viable. But this has a drawback where you cannot do single cell analysis. You can only do single nuclei analysis, because you will have to lyse the cell and take the nuclei out. And what happens when you do that is you lose your surface proteins, and you cannot kind of do the whole site-seq analysis. The other thing is cryopreservation in medium. This is kind of a long-term storage, which is more than 72 hours, which is less than 72 hours, I'm sorry. And you can preserve your, and again, this is less than 72 hours. So you need to be sure that you process your sample right before 72 hours. And you would preserve your cell markers in this, and you can do single cell analysis in this. This is a very viable pilot method. And if you're doing single cell, it's very important that you have a viability and cell count which is more than 80%. So if you have less than 80% of cells which is viable, you're going to end up with RNA, ambient RNA. So in case you have more dead cells or debris, what are the steps? You kind of do multiple washings, you kind of do filtration with microfilters like max stainers, which is like 30 micron stainers, so that the debris are removed. Or you do something like beads, or you do FACS, where you kind of do an enrichment, a depletion with a magnetic bead. So this is necessary. If not, it's really not recommended that you go for single cell because sequencing, because it's really expensive, and if you don't have a good RNA quality and a cell count, you're not going to end up with the good data. Okay, let's dive into what typically you can do with single cell RNA sequencing. So there's two steps. Like the pre-processing is what we were like talking about sequencing. So when you sequence, you get a matrix, which is the gene and the cell count. And these are just the steps to follow after you get your data. You need to see the quality of the cell count. You need to see how the sequencing depth is. Even during sequencing, there can be trouble where the sequencer kind of didn't take the whole sample and you don't have a sequencing depth. So you need to do the QC, and it's very important. The other thing was the batch effect. Like you need to really control for the batch effect. In case you have multiple samples, multiple batches of samples, it's very important to control the batch effect. And one more thing is normalization. This is kind of a basic step because we get the gene counts in hundreds and thousands, and it's very important to normalize it so that all the counts are in an normal distribution. So either log normalization can be done, or you can do scale normalization. The other thing would be once you have the pre-processing data done, you could work on the downstream analysis, like okay, I'm not going to go deep into the downstream analysis. I'm just going to say that there is clustering and there is trajectory analysis and differential expression. Uh, I will be diving deep into these topics um, in the next slides. So let me start with an example of what single cell analysis kind of works on.
Dr. Rita voice is not audible. You are on mute.
Do you hear me now?
Yes, yes,
I'm so
yes. We
sorry
can
about
hear
that.
you.
I have no idea
It's
how
okay.
to get you.
Yeah, yeah, yeah. No issues. You just again share your screen.
Yes.
Yes.
Bye. Can it? Can you let me know from where, when did the voice go off? I can start from there again.
Next, next.
Next.
From here actually.
Okay, 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 got it.
After this you completed.
Okay,
Downstream. Next slide.
okay,
Yes,
okay,
yes.
perfect. I'm sorry about that, guys. I didn't realize that my internet had some glitch on it. Okay, so I was just telling you guys about the tool which 10x Genomics gives. They have their own browser, which has a graphical interface called, and it's named as Loop Browser. And they have the cloud where you can try to upload your data and you can do the analysis. So what they give you is, this is just example of what the analysis they help you do, and this is they help you examine the gene expression. This is kind of the gene expression analysis where they kind of have looked into the CD3 and CDE gene expression, which is just the basic mRNA expression. They also help you do the T cell and the cell type deconvolution, which is like they help you to analyze what is the genetic marker, what is the cell types you can analyze. In specific for this whole topic, I'll just be showing about T cells. And you could see that this red and this blue is, this yellow is what is the T cell markers, and these are the clusters for T cells. So if you could see the next image, this is the surface marker identification. So the next step they took to do is, they added surface markers, which is like CD45A, which is nothing but a cell surface marker for immune cells. And they tagged this onto the mRNA to show that there is expression of immune cells. So you could see that clearly the T cells, the B cells, and the immune cells, like the basophils and eosinophils, all these are dark red in color, which shows more expression than the other cell types, which is light in color, light red. The other thing which they help you to analyze is the gene expression along with the T cell and B cell markers. Like you could see the T cell over here has, which is violet cluster. Um, this kind of shows you the T antigens, like the Tregs, the T cells and the B cell receptors, and the VDJ chains. V is okay, it's just the segments for the antigens, which is V, D, and J. In case you have a alpha chain, you'll have V and J, and in case you have a beta chain of the TCR, you'll have the D chain along with it. So, so the browser helps you kind of analyze it, but the only glitch is that you cannot kind of upload a huge file at a moment, because there is a limit for the cloud. And that's kind of the drawback where you have to choose for programming. This is a paper which we recently published in Cells in 2022. It's about the functional and phenotypic characterization of cyclic 6 on mast cells. So we did this analysis on two cohorts. When I say cohorts, it's just like two different patient population. And we found that the cyclic 6 was a gene marker, genetic marker, which was specifically found on human mast cells in both the cohorts. So this data is just going to prove you the same. Um, if you could see the image on the left, which has A, it's just a cell type identification future plot, which kind of shows that the mast cells, what are the different types of cells, like the epithelial, the mast cells, the T cells, the endothelial cells, the B cells. This was basically the cell types found in the sample. And the B image is just going to tell you the different markers. If you could see the mast cell markers, we were looking at TPSAB, which is like the most expressed one, which says that these cells has been mast cells. You have different other genetic markers for each of the cell types. I'm not going to dive deep into that. And this image is called a heat map, which kind of shows the cyclic 8s are highly expressed in mast cells. So red is the highest expression, which is upregulated, and it shows that it's highly expressed in mast cells. The image below is just a similar image, which says this group is the mast cells, and it has cyclic 6, and the below is cyclic 8. So it shows that cyclic 6 expression is more than cyclic 8. These two bar plots are nothing but the cyclic 8 expression based on the disease type. So based on whether the disease is active or inactive, it just shows how much of expression was there on the patients with active disease and it's compared between cyclic 8 and cyclic 6. The same type of analysis was done in other cohort. This was between two different hospitals, one in Cincinnati and other was Lurie Children's. So we did the same analysis and we found that we had this TPSAB marker, which is a genetic marker for mast cells, which was present in even our data set. And also cyclic 6 was highly expressed at cyclic 8. And the same pattern was followed in patients with active. You could see that they had more cyclic 6, which is the green marker, the green colored plot. This is called a violent plot, and it's just comparing the patients who have active disease and who had inactive disease after the treatment and between the control patients. This whole test was very. This whole paper was suggesting that we use single cell, where we were able to prove that cyclic 6 was a genetic marker which was present in the diseased patients, and it was a marker from mast cells, and mast cells were highly regulated in the patients who were suffering with EOE. I've given you a brief of what single cell is and all this. I'll dive into the computational part to tell you what type of tools we use and what are the pipelines, the bioinformatic pipelines we use for the analysis. So the major, for single cell RNA, the most used tool is Surat. They have all the pipelines for site-seq with single cell, and also for TCR and PCR with single cell. They have the whole pipeline developed. And the second tool we use is single R for the annotations of the cell markers. You can do a literature survey saying the tryptase biomarker is for mast cells, but it's going to take you a long time. So they have this whole annotation tool called single R, which has previously annotated databases where you can pull the genetic markers from that. The other tool is monoclone. This is kind of the next step for single cell. It's called trajectory analysis or lineage composition, where it kind of tells you what is the path of the, suppose you have a T cell, what are the subtypes and what is the lineage? How is one cell type triggering or activating or suppressing the other cell types, whether it's a Tregs which activate the helper cells, or is it the other way around? So all these type of trajectory analysis or lineage, you call them, you could do it in monoclone. The next thing is the scatter. We use this for the pre-processing, the initial step like the quality control, the normalization, and all. And you can also visualize using scatter. All this is done in RStudio. You could use Python also. You have Scanpy for Python, but I use more of R, so I'll be giving you a description of all the R tools. Um, to start with the data analysis, the first part is the filtering and the pre-processing, which is the quality control. So it is very important that you follow these steps, because if this is not followed, you're not going to get a genetic profile which matches the cell type. It's going to be a profile which has mRNA, which has ribosomes, or which has contaminations or RNA. So the first step would be trying to remove the artifacts and trying to see the quality control. So when I say artifacts, it's nothing but the dead cells, the empty wells. Like, you have those gel beads which can be empty without any cells, or which can have 2 cells or 3 cells within a bead. So when you get your data would show all this, whether it's a dead cell or it's an empty cell or it's a doublet. And also, it's very important that you remove the mitochondrial and ribosomal contamination from your data, because that's not going to uh, lead to any pathway analysis. Like, your mitochondrial or ribosomal gene is not going to give you any significant genetic marker. This is just to show. The plot in the left is just showing you the number of genes and the number of UMIs, which is the cells. And you need to have a correlation which is more than at least 0.9, saying that your QC is good. And the plot in the right is nothing but the genes and the UMIs, showing that you have about 5,000 genes, and you have about 20,000 cell counts. The next step would be, once you have the QC done, the next step is filtration and processing. So the next step is normalization. So in normalization, it's very important that you normalize your data, because you might have values which are really in thousands. So you need to normalize the data so that you have a normal distribution and your counts are within a range where it can do the differential expression. So few of the normalization steps are, you can either do, the most commonly used is log normalization. In case for some reason you're not able to, log normalization doesn't work for you, then you can go for scale or z-score or median normalization. The next thing is variable genes. To find the variable genes, you need to set a threshold. So in this plot, you could see that anything which is above a range of 3 to 4, it's not marked, because we don't want genes which are highly variable. And it's not going to be a gene which is regular, which is among all the cells. It might be a gene which is kind of really highly expressed due to doublets or triplet, or due to some other debris. So we kind of have a threshold saying anything which has an average expression of 2 or 3, go with those genes. Similarly, there are many other steps you can do. You can look into the median, you can look into the dispersion. It's more important that you have a statistical significance. So in this, we are looking into the dispersion, and we're kind of seeing how well the genes are dispersed. The other thing is reducing noise. I think I touched it in the beginning, which is batch correction. This is one major thing which is very important to reduce noise, and this can be done by the cell cycle. So the image in the left is just telling you about the technical variability. Like, if you have a very huge technical variability, you need to control for that. And the right is just an image where they have control for the noise. Similarly, the batch correction, the plot over here shows that the samples are batched as clusters. So you cannot have these samples separately batched. You need all the samples to be merged together, has a merged sample. So you cannot have sampled clusters. And it's very important to correct for that. Once you have the QC done, the next step is doing the dimensional reduction, where you kind of come up with a PCA plot or a UMAP or a TSC plot to show the clusters. When I say clusters, it's just like the genes which belong to one cell is all going to cluster. One cell type is going to cluster together. So if you can see this PC1, you could see that there's two clear clusters between these cells. So it's very important that you have, when you're starting the analysis, you take a PC which doesn't have this variation. And for if you could see PC9, it doesn't show a variation between the samples. So it's very important that you go into a PC dimensional reduction, which is not showing a distinguish between the samples. And in PC1, this is just showing you how the distinction. In PC1, you have markers which are kind of distinguished, but when it comes to PC2 and PC3, the variability would be much more lesser. So for this analysis, we would go with PC9. The next thing is you can either do PCA, which is like a linear method, which is the most basic, or in case you feel like even while using a PC of 9, I'm not getting a proper cluster, then you could use a nonlinear model, which is Disney plots or UMAP. Disney plots and UMAPs are good because they account for the Euclidean distance between the points, which doesn't is not accounted in the PCA analysis. So if you could see that in the Disney plot, you can see different cluster groups, whereas in the PCA plot, you could see that there wasn't a clear cluster of the cells. Um, for the nonlinear method, there are two methods: either it's Disney or UMAP. Whichever you feel gives you a good result with your cell type, you could go with that. UMAP is much more comfortable, because you can go for the subclustering, and it kind of has a good. It gives you a good plot than Disney. And this is just an example showing the UMAP, and this is just a TST monophage, a mononuclear biomarker, which kind of shows that this group is this group is the cell type which expresses CST3. The next thing after the dimensionality reduction is the cell clustering. So during cell clustering, there are some parameters which you need to follow. You need to give what the resolution and what the clustering type you need. And this is also one thing which would depend on the data set. So for some people, they would have a data set which is like really cell type which is very closely related, like suppose T cell, Treg, T helper cells, Th1, Th2. So when you have such cells which are very closely related, you're going to have a problem if you're going to use hierarchical clustering, because it's not going to cluster them as subtypes. So in such cases, they go for the other two type. They use expectation normalization or k nearest neighbors. This kind of helps you kind of to do a better clustering. This is your original data set, but when you do k-means or EM clustering, you could see that you have a totally different cluster. You don't have all these outliers like which are surrounding the cluster. It's properly clustered, and you have three exact groups. This is more like a trial and error based on your data set. You could like choose whichever gives you the best clusters. The other thing you need to choose, we choose the PC, which is 9. The other thing is the resolution. So the resolution in Surat is always 0.6 to 0.1. This resolution is based on the cluster group which you want. It's whether you want a bigger cluster or you want smaller clusters. So the more you increase the resolution, the more clusters you get. There are 0.6 might have like 6 clusters. So if you go to 0.8, then you might have 8 or 9 clusters, and the max you could go is 1.2. And above that, you're going to have the more smaller clusters, which doesn't really make any sense. This plot is nothing but an elbow plot, which kind of helps you determine the PC. You can do a PC, which we saw earlier, to show that 9 was the cluster, or you can also determine it based on the standard deviation, where after 9, you can see that the 10 is almost like the standard deviation is very less. So we go with a PC of 9. The next thing would be, once you have the clusters, you could see a heat map. So for this clustering, we took a cluster of 1 is to 15 PCs and a resolution of zero point 0.8, and we found that 9 clusters were there. And for each of the clusters, you could see that there is a group of genetic markers, which kind of tells you that it is an individual cluster. So, and if you could see the 8th and 9th, it still has a small cluster, but not a lot of cells in it. But it's still a good cluster. This is just a visualization of the heat map to show what the different clusters are. And you could see that the 9th cluster, though it is a small cluster with very less number of cells, it still is a cluster. It's still a pretty good cluster. Once you have these clusters, the next thing is you need to annotate it to find what these clusters are. So you have the genetic markers for each of these clusters. You could do a single R to find what the clusters are, or you could go for a literature search trying to see what of the CD4 is a T cell biomarker, tryptase is a mast cell biomarker, CD8 is an CD8 T cell marker. You can do a literature search and find it, but that's going to be really hard, and it's much more easier to use a tool like single R. MPO, this is a monocyte biomarker, and you could see that it is highly expressed in cluster 2, cluster 3, and cluster 7. So as you saw the plot earlier, cluster 2, cluster 3, and cluster 7 is this whole chunk. And you could see that this whole chunk is MPO. So it has a higher expression, which is a darker blue than the rest of the cells, showing that this is kind of a marker for mononuclear cell types. The other thing is CST3. This is a protein marker for the dendritic cells. And you could see that it is highly regulated in cluster 9. And you could see this small chunk of cluster was cluster 9. And this kind of cells that CST is in biomarker for dendritic cells. So with this technique, you can try to annotate all the different cell types to say that these genetic markers are for these cell types. And in this analysis, we were able to come up with 9 different clusters saying these were the NK cells, and these were the dendritic cells, and these were different cell types. So this was based on purely the genetic marker, which was derived from the cells. Once you have this, it doesn't finish over here. You can take this further to do different analysis. Like for bulk-seq, you do differential expression to see what are the differentially expressed genes between the disease sample and the control sample. So you could do the same with the clusters which you get, because you have the genetic markers for each of the clusters. You can see how they vary with the cell types, with the disease status, and all those things. I spoke about trajectory analysis and linkage. This is a very interesting study because it gives you the projector of the cells, which cell comes after which cell type comes after which cell type. Like it just gives you the suppose you have the myeloid cells, it gives you the progenitor of the myeloid cells. The other thing is differential expression. You could see the differences between the disease type or the activity of the disease, or any of the clinical data. You could try to put it into this data, and you can say the difference between the expression. The other interesting thing is, once you have this differential expression, you don't have to just say these are just the genetic markers I have. You can go one step ahead and say because of these genetic markers, I have these pathways which are regulated, and these pathways are in the disease patient. And you could also say what are the other genes which are enriched, along with, suppose you have, for example, you have, what is it? Uh, yeah, you have a TPS-AB. You could say what are the other genetic markers along with the mast cells, the trypsin biomarker, which are correlated with the marker. So it gives you a gene set enrichment analysis, to which also helps you to find the genetically enriched pathways. The other thing is, as I already told you, the functional analysis or the composition analysis. You could check it with different conditions, different cell types, different frequency of cell types. The last one thing which people are trying to go one more step ahead is the pseudo analysis. Pseudo analysis is nothing but the time series analysis of the cell cycle. So what is happening to the cells? Is it in the G phase, or is it in the M phase, or is it moved to the next cell fate, or is the cell differentiated? So people have started using the single cell data to do the pseudo time analysis to know what the cell differentiation is. Uh, Last year, 10x came up with this whole new concept of spatial organization. They call it spatial transcriptomics. Spatial transcriptomics is nothing but you have your tissue on a slide. So with a slide, you could just do staining, and you could say that this genetic marker is there, this antibody is there or not. But 10x Genomics has taken a single cell to a really next level, where they have tried to do genomics on these slides. So if you have have a tissue slide, you could do single cell on the tissue slide. You could do the analysis using the microscope, and then you could see what are the biomarkers are there on the microscope, and then use GenM technology to remove those biomarkers from the slide and do the sequencing. So it's kind of an way, whole next world where with just microscopic slides, you could do single cell analysis. The other thing which they have they had come up with is the T cell and B cell antigen receptors, and also the cell to cell communication, which is the site-seq, which is the cell surface markers. So these are all the recent approaches which are still under development, and there are a lot more. It's kind of people are trying to develop a lot more approaches with single cell. Um, so you could put it on your chat what you have learned from this whole single cell analysis, whether it was the tool or whether it was the heterogeneity of the cell types you learned or anything. Before that, I could like to go for some data analysis just to show how our programming works. Okay, this is. Okay, so this is just the Surat tool, which actually is loaded into R programming. You first have to load the library, and this is actually the design experiment, which has the cells. And in this particular experiment, in this particular analysis, I'm just looking for the CD41 cells, which is actually the myeloid cells. I'm trying to look into the subtypes of the myeloid cells. You kind of load the data. You look into what is the length of the data, like how many genes you have, and how many cells you have. So this kind of gives you the violent plot of how many genes you have and how many cells you have, via which after the quality control. This is just showing you the correlation between the cells and the genes, which is like 0.98, which is a good QC. And then you have the step where you try to filter the highly variable genes. As I told you, you could use anything like mean, median, expression, dispersion, anything. And in this, we have taken log BMR, which is log variance median ratio. You could either go for log scale or you could go for any other normalization. It's still fine, like the Z normalization or median normalization. So it just depends upon whichever you get the best results. And the next step is scaling the data. And we have the elbow plot, which kind of shows you what are the PCs, which kind of was good. I think for this data we had 15, which was after which there's not much standard deviation. This is just a PCA plot, but we just skip the PCA because we don't get the better clusters. And this is the Disney plot where we had all the different cell types clustered well. This is just in comparison to show that the Disney plot looks good when it comes to clustering than the PCA plot. And then you have your heat map, which has different clusters, 1 to 9, and each of the clusters have the genetic markers. Like the cluster 9 had CST3, which was the dendritic cell. So it shows that this cluster is kind of heavy with the dendritic cells marker. And it has an expression which shows yellow, which is highly expressed in this case. You have the violent plots, which kind of shows you it's the same in the heat map. You cannot see the expression. In this, it just shows you the expression. It's expressed. The dendritic cell marker CST3 is expressed at a level at an average expression of 6. Similarly, the MPO, which is a monocyte biomarkers, are expressed within the 2, 3, and 7 clusters. It's highly expressed. This is the same type of plot, but it's just a future plot, which kind of which kind of shows you the visualization based on the clustering, just like this one. So it just is a similar plot. And then you have the clusters labeled, and the next step would be to label these clusters based on these cell markers. So this is about all the single cell and the bioinformatic tools. I could take questions if you guys have any.
Yeah, thank you, Dr. Retha. It was very informative and very beautifully explained session, actually. So let's take some questions. First question is how to measure cell viability.
Okay, so you can measure cell viability using the cell counter, or you can just do a Trifon blue staining or AOPI staining and see the number of viable cells and the dead cells. You could just do it on your microscope. Yeah.
Okay, next question. The next one is can these genetic markers use for personalized medicines?
Surely. So the whole point of single cell is to develop immunotherapeutic drugs, genetic marker drugs. So this is kind of like an initial step. And I showed you one of the paper where we are kind of working on Siglic 6, which is an activation marker for mast cells. So the whole idea is to find these genetic markers so that over time we can either knock them out or we can kind of suppress them to see if the reduction in the activity kind of helps the disease status in the treatment. Yeah.
Do initial fast QC analysis are required for single cell sequence analysis?
So you could do all this so fast QC is for bulk seq analysis. For single cell, you have different other analysis, which is like the Hamming distance, and it's very important that you have a Hamming distance of 1 between the genes. And QC is kind of something which is very important, but fast-Q analysis is done for the bulk seq analysis, where you have just the genetic markers.
Yes. Yes. Next is which aligner is preferred for cis RNA?
You could so it's your preference. You can use either stop hat or star. Both are good. I've used personally star, and I think my analysis was pretty good.
What is the advantage of sgRNA compared with the bulk RNA sequence analysis?
Okay, this is like the the whole point which I told you in the beginning. For bulk seq, you don't get the cell types, you get only the genetic markers. So you don't know from which cell is the genetic marker coming from. So it's very important to know what is the cell type you're looking at. So is it an immune? So all these immunotherapies are mainly targeted for immune cells, and it's very important to know from which cell type, like whether it's a T cell or B cell, your genetic marker is coming from. So bulk seq is kind of like a preliminary analysis. You could just get to know these are the genetic markers, but it is very important to go for the single cell analysis to know from which cell is the genetic marker coming from.
I think we have done with all the questions. Dr. Rita, thank you for such a wonderful and well explained session, I must say. So, thank you once again for connecting with us, and we was very it was very wonderful to have you with us. Actually, we are grateful that you could have joined us today. So, thank you all. We hope that to see you all there in our next forthcoming sessions of this Women's Day webinar special webinar series, actually. So stay tuned with us. Thank you. Thank you all.
Thank you, everyone.